Today we are going to continue our journey, our interesting journey through the course of normal physiology. And we are going to begin to focus on the nervous system and to its components at a microscopic level, as well as the physiology, not so much where things are and what they look like, but really how they work and how all of this system gets integrated. The nervous system is a very complex organ system. In Peter Kramer's book, Listening to Prozac, a pharmaceutical researcher is quoted as saying, "If the human brain were simple enough for us to understand, we would be too simple to understand it." That quote is from the early 90s. In several decades since, progress has continued at an amazing rate within the scientific disciplines of neuroscience. It is an interesting conundrum to consider that the complexity of the nervous system may be too complex for it, that is, for us to completely unravel. But our current level of understanding is probably nowhere close to that limit. One easy way to begin to understand the structure of the nervous system is to start with the large divisions and work through to a more in-depth understanding. In other chapters, the finer details of the nervous system will be explained, but first looking at an overview of the system will allow you to begin to understand how its parts work together. The focus of this chapter is on nervous neural tissue, both its structure and its function as well. Our first question is divisions of the nervous system. The nervous system can be divided into two major regions, the central and peripheral nervous system. The central nervous system, CNS, is the brain and spinal cord, and the peripheral nervous system, PNS, is everything else. The cranial, spinal nerves, etc. The brain is contained within the cranial cavity of the skull, and the spinal cord is contained within the vertebral cavity of the vertebral column. It's a bit of an oversimplification to say that the CNS is what is inside these two cavities and the peripheral nervous system is outside of them. But that's one way to start to think about it. Actually, there are some elements of the peripheral nervous system that are within the cranial or vertebral cavities. The peripheral nervous system is so named because it's it's on the periphery, meaning beyond the brain and spinal cord. Depending on different aspects of the nervous system, the dividing line between central and peripheral is not necessarily universal. Nervous impulses travel in two directions, and each direction has a separate set of nerves. efferent or descending nerves send impulses away from the brain. These are also called motor nerves. And the sensory impulses that ascend the nerves to the brain are called afferent impulses and move along afferent nerves. Efferent impulses produce action in distant organ, but afferent impulses deliver information from peripheral nerves to the brain. The integrative function analyzes information input from afferent impulses, files these data, and stimulates action in response to sensory input. Kinds of nervous tissue and their functions. First is neurons, very significant parts, general, major parts of nervous system. Neurons are nerve cells. They initiate and relay electrical nervous impulses toward and away from the brain. Neurons have three basic parts: the cell body, dendrites or trees, which receive incoming messages, and axons, which deliver outgoing messages. Neurons vary in their function. They can be sensory, motor, or integrative neurons. Neurons vary in size and shape and in the number of axons and dendrites that they may have. Due to structural differences, neurons can be classified into three major groups. First group is bipolar neurons that have one axon and one dendrite. Unipolar neurons with one axon and small dendrite, and multipolar neurons that have one axon and many dendrites. Sensory neurons. Sensory neurons, they are afferent and they carry impulse to central nervous system. Second are interneurons. They link neurons as known as association neurons. They located in central nervous system. And motor neurons. Motor neurons are multipolar neurons and they carry impulses away from CNS to periphery. Carry impulses to effector organs. So nerves can have any arrangement of dendrites and axons, including one input to many outputs. Divergence. Many inputs to one output. Convergence. And one input to one output. Some axons are myelinated or insulated for rapid impulse delivery. These axons display saltatory, jumping conduction. Other axons are unmyelinated, and what's myelin? Myelin sheets develop as a baby growth. Disease can interrupt this process. Myelin sheets consist of Schwann cells, fat cells. There are different types of cells in nervous system. Neuroglia or nerve glue. They are connective. Neuroglia are connective nerves that hold the structure of the nervous system together. These cells nurture the neurons. They are much more numerous than the neurons and are regenerated over time. Sorry. Because they can regenerate, Their stem cells, not the neurons, are the source of most cancerous cells in brain cancer, it's said. So there are different types of these cells. First are microglia. They are located in CNS, and they are phagocytic cell. Then astrocytes, located also in CNS, scar tissue, mop up excess ions, etc. Induce synapse formation, connect neurons to blood vessels. They are all microglia cells. Second type of cells are oligodendrocytes. They are located in central nervous system. They are myelinated cell, located also in ependyma or ependymal cells, ciliated and line central canal of spinal cord, line ventricles of brain. These were neuroglia cells. Our next question is reflexes. Reflexes are rapid, automatic, unlearned motor response to a stimulus. It usually removes or opposes the original stimulus. Monosynaptic reflexes, first type of reflexes. For example, the stretch reflex, which responds to muscle spindles. Spindles is the simplest with only one synapse. The best known stretch reflex is probably the knee jack reflex. You know it.
Involve me tapping just with this small tendon hammer on parts of your legs, okay? So I want you to let your legs go floppy for me. Okay, let me take the weight. Okay, same on the other side, just let it go floppy, let me take the weight. Okay, okay. Now if you can bend this leg and just let it flop out to the side for me. And just relax your ankle, just let it go floppy for me. That's it. Good. Same on the other side for me. Pop it out to the side. Okay.
It's simple reflex. Right in a reflex arc, stimulus activates a sensory receptor. Sensory receptor is the first part of reflex arc. In action potential travels down an afferent sensory neuron and bring information to the central nervous system. Information processing occurs with the interneuron. An action potential travels down an efferent neuron. And then last part of the reflex arc is the effector organ. Effector organ responds to a stimuli. So first part of reflex arc is a receptor. Second, afferent neuron and afferent nerve. Next, central nervous system or center of this reflex. Fourth part is motor neuron or efferent neuron and effector organ. Complex reflex, polysynaptic reflex with at least one interneuron, one or two or thousand interneurons, are slower than monosynaptic reflexes but can activate more than one effector. Withdrawal reflexes, like the flexor reflex, move a body part away from the stimulation, like touching a hot stove. Reciprocal inhibition blocks the flexor's antagonists to ensure that flexion is in no way interfered with. So our next question is the synapse. synapse is a very significant, very crucial part of the nervous system. Synapse is a junction between a neuron and another cell, nervous cell, neuron, or muscle cell, or secretory tissue cell. Occurs because of chemical messengers called neurotransmitters. Communication happens in one direction only. Between a neuron and another cell type is a neuroeffector junction. Such as the neuromuscular junction or neuroglandular junction. The neurotransmitter acetylcholine is a general neurotransmitter in the nervous system. It activates cholinergic synapses in 4 steps. Remember, please, 4 steps of activating cholinergic synapses. First, action potential arrives at the axon terminal. Acetylcholine is released from vesicles that located in the presynaptic membrane and diffuses across synaptic cleft between presynaptic and postsynaptic membrane. Acetylcholine binds to receptors and triggers depolarization of the postsynaptic membrane. Acetylcholine then is removed by acetylcholine esterase. There are other important neurotransmitters except acetylcholine. For example, norepinephrine, adrenaline or norepinephrine in the brain and located in the brain and part of the autonomic nervous system is found in adrenergic synapses. Dopamine and serotonin are CNS neurotransmitters, at least 50 less understood neurotransmitters. For example, NO and CO are gases that act as neurotransmitters, as mediators. There are excitatory and inhibitory synapses. Usually, acetylcholine and norepinephrine trigger depolarization. They are excitatory transmitters in excitatory response with the potential of reaching threshold. Usually, dopamine and serotonin trigger hyperpolarization. They are inhibitory transmitters and inhibitory response, making it farther from threshold.
The nervous system is composed of billions of communicating cells called neurons. Neurons conduct electrical signals or action potentials along the cell's membrane, along the axon to the terminus, where synapses are formed. The synapse consists of the membrane of the presynaptic cell, a synaptic cleft, and the postsynaptic cell membrane. Since an action potential can only propagate along the neuron's membrane, a chemical known as a neurotransmitter must bridge the gap between the adjacent cells. The presynaptic cell releases the neurotransmitters into the synaptic cleft, where they will move to bind the receptor proteins in the postsynaptic cell membrane. When the action potential arrives at the synapse, it opens voltage-gated calcium channels, resulting in an inward flow of calcium. The calcium binds to the vesicular membrane. This binding activates the vesicles to fuse with the presynaptic membrane. As fusion occurs, neurotransmitter molecules are released into the synaptic cleft. Once in the cleft, the neurotransmitter molecules diffuse throughout the interstitial fluid in the cleft. Some of the neurotransmitters will cross the synaptic cleft and bind with receptors in the postsynaptic membrane. This binding produces an effect in the postsynaptic cell by opening the protein channels and allowing the flow of ions into and out of the cell. If the effect of the neurotransmitter excites the postsynaptic membrane, an excitatory postsynaptic membrane potential, also known as EPSP, is generated. This localized depolarizing signal generally causes long-distance signals, action potentials, in the dendrite of the postsynaptic cell. If the effect of the neurotransmitter inhibits the postsynaptic membrane and inhibitory postsynaptic membrane potential, also known as an IPSP, is generated. This localized hyperpolarizing signal generally prevents the formation of long-distance signals in the postsynaptic cell. Neurotransmitters influence the postsynaptic membrane electrical state for only a short time and are removed from the synaptic cleft in one of three ways: diffusion away from the receptors, enzymatically degraded, or recycled by the presynaptic cell. Some of the neurotransmitter molecules diffuse out of reach of the neurotransmitter receptors. Some neurotransmitter molecules are removed by enzymatic degradation. Some neurotransmitter molecules are transported back into the presynaptic cell by neurotransmitter transporter proteins, where they are recycled. An example of a specific neurotransmitter that functions in these ways is serotonin. Some serotonin will cross the synaptic cleft and bind with the specific serotonin receptors within the membrane of the postsynaptic cell, producing an effect in the target cell. Serotonin will eventually unbind from the receptors and either diffuse out of the synaptic cleft and be lost, or be captured by the serotonin reuptake transporter in the presynaptic membrane. This protein complex will transport the biologically active serotonin back into the presynaptic neuron for repackaging and re-release. Acetylcholine, the first neurotransmitter discovered, also diffuses across the synaptic cleft and temporarily binds with its specific membrane-bound receptor on the postsynaptic cell, affecting the cell's electrical state. Some acetylcholine in the cleft will interact with the enzyme acetylcholinesterase, which will hydrolyze the acetylcholine molecule into acetate and choline. Acetate diffuses away, but choline interacts with the presynaptic membrane-bound transporter to be recycled in the presynaptic neuron into new acetylcholine. Unlike serotonin, which is intact and biologically active following its uptake into the presynaptic cell, new synthesis of acetylcholine is necessary, with the recycled choline as the precursor.
Hi there, my name's Andrew. I'm one of the final year medical students. Could I just confirm your name and age, please?
Yeah, my name's Robert Brown and I'm 21.
Nice to meet you, Robert. Today I've been asked to examine your cranial nerves. These are the nerves that supply your face and your neck. Would that be okay?
Yeah, that's fine.
Brilliant. Are you in any pain at all?
I'm in no pain.
Okay. First of all, Robert, I'd just like to check, have you noticed any change in your sense of smell at all?
No, I haven't.
Okay. Now I'd like to test your vision. Would that be okay?
That's fine.
I'd now like to assess your visual acuity. Do you usually wear glasses, Robert?
No.
Okay. If you could cover one eye with the palm of that hand, and starting at the top, I'd like you to read down as far as you can on the Snellen chart in front of you.
EHN DFN PTXZ UZDTF DFNPTH PHUNTDZ NPXTZFH.
Fantastic. And if you could cover the other eye and do the same.
EHN DFN PTXZ UZ DTF DFNPTH PHUNTDZ NPXTZFH.
Brilliant. I'd now like to assess your pupils. This will involve shining a bright light into your eyes. Would that be okay?
Yes.
Brilliant. If you could just pick a point on the wall in front of you and keep looking at that. That's great. I'd now like to swing the light between your eyes. If you just keep looking at the point on the wall in front of you. Finally, I'd like to assess the ability of your eyes to focus. If you could look at the point on the wall in front of you. And now look at the pen. That's great. Robert, I'd now like to do some specific tests of your vision. If you could just keep looking into my eyes, I'd like you to point at the finger you see moving. Brilliant. I'd now like to test your visual fields. If you could just cover one eye with the palm of that hand for me, I'm going to cover the opposite eye. Let me know when you see my finger moving. Keep looking into my open eye.
Yes. 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 Yes.
Cover the opposite eye for me. Same again.
Yes. 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 Yes.
Brilliant. I'd now like to assess the movements of your eyes. First of all, have you got any double vision at the moment?
No.
Okay. Do let me know if you have any double vision throughout the exam. But I need you to keep your head perfectly still, and I'd like you to follow my finger. Did you have any double vision at all?
No.
Okay, fantastic. Okay, I'd now like to assess the sensation on your face. First of all, I'll start with a piece of cotton wool and then a small neurotip. Would that be okay?
Yes.
Brilliant. This is what the cotton wool feels like. Can you feel that?
Mm-hmm.
Brilliant. If you could just close your eyes and let me know when you feel the cotton wool on your face.
No. 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 No.
And did it feel the same on both sides of your face?
Yes, it did.
That's great. I'm now going to assess it with this neurotip. Can you feel that?
Yes.
Fantastic. Again, if you could close your eyes for me.
No. 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 No.
And again, did it feel the same on both sides?
Yes, it did.
That's great. Okay, I'm now going to feel some of the muscles around your face now. Just clench your jaw for me and relax. Clench your jaw for me and relax. Open your mouth and don't let me close it. I'm now going to assess a reflex, if that's okay. What I'd like you to do is just relax your jaw down slightly for me. I'm just going to place my finger on your chin. Fantastic. So I'd just like to assess some movements of facial expression now, Robert. First of all, can you start by raising your eyebrows? Great. And now if you can close your eyes as tight as you can and don't let me open them. If you can puff out your cheeks, don't let me push them in. You can purse your lips for me and bare your teeth. That's great, thank you. 2 quick questions for you, Robert. Firstly, have you noticed any change in your taste at all?
No.
Okay. And secondly, have you noticed that your hearing sounds louder than usual?
No.
Good. Robert, I'd now like to assess your hearing, if that's okay. To start with, I'm going to place one hand on one ear and I'm going to whisper a number or a word into the other ear. I'd just like you to repeat it. Okay?
66. 79.
Fantastic. And just the same on the other side.
83. 97.
That's great. So I'm now going to use the tuning forks to assess your hearing. First of all, I'm going to place the tuning fork on the bone behind your ear. I'd like you to let me know whether you can hear it and when you stop hearing it.
Okay.
Can you hear that?
Yes.
Let me know when it stops.
It stopped.
And can you hear that?
Yes.
I'm just going to do the same on the other side. So I'm now going to place the tuning fork in the middle of your forehead. Can you hear that, Robert?
Yes.
And is it loudest in one ear or the other or in the middle?
In the middle.
That's great. I'd now like to assess the nerve that assists with your balance. And now I've got you standing up. What I'd like you to do is close your eyes and I'd like you to march on the spot for me. That's great, you can stop now. Robert, if you could just cough for me. Finally, if you could just take a sip of this water. And when you're ready, swallow. So now I'd like to just assess the power in your shoulders and your neck. Robert, if you could just start by shrugging your shoulders for me and don't let me push them down. Fantastic. Now if you could turn your head to the right against my hand and don't let me stop you. Fantastic. Turn your head to the left against my hand and don't let me stop you. That's brilliant. So I'd now like to examine your tongue. First of all, if you can just open your mouth for me. That's brilliant. And if you can stick your tongue out. Fantastic. Now if you can just press your tongue into my fingers. And the same on the other side. Today I did a cranial nerve exam on Robert Brown, a 21-year-old male. On examination of cranial nerves 1 to 12, there were no abnormalities detected. To complete my examination, I'd like to do a motor and sensory exam of both the upper and lower limbs.
Thank you. That's it. Welcome to my next lecture next week. Bye. Good luck.